All right, now I start sharing my slides. Okay, can you see my slides? Okay, now I show full slides and start. All right. Hello everyone again. Today talk is about methodological aspects of human research with transcranial electrical stimulation, but most specifically with TDCs, which we start. So I have nothing to disclose financially, but I serve as editorial board in some journals, as you can see. I like to talk about a little bit my background because it's important that how I got interested. So my background is in psychology, mostly clinical psychology. Then I got interested in my master thesis. My master thesis was about application of transcranial direct current stimulation in depression, and that was the point that was a turning point in my academic life, and I got interested about brain, neuroscience, and then I changed my path. Although now most of my works still are clinical, as you will see soon. I did my PhD in cognitive neuroscience in the International Graduate School of Neuroscience, but in Germany the system is like that you are in one university, but you can do your project elsewhere. So my project was done in Leibniz Institute in the neuromodulation research group, which is supervised by Professor Michael Nietzsche, who probably you should know if you are in the field of non-invasive brain stimulation. And my thesis was about the impact of circadian factors on cortical excitability, neuroplasticity, and cognition. Today, I'll also show you some slides about some of the some findings of my thesis, which are related to our topic. Okay, this is about me. I like to also know about you, but this is not possible. Okay. This is the overview of talk. First, I talk about general, very brief introduction of non-invasive brain stimulation and mostly transcranial electrical stimulation, because this is one major part of non-invasive brain stimulation. Then talk about TDCs, how it works, the physiological effects, and then from this part, our discussion begins about the factors that affect the observed effect of TDCs. These factors include inter-individual variability factors, stimulation parameters factors, some factors that we can control in the experiment, and also some experimental aspect. Briefly, I cannot cover all of it. This is just very brief touch of these topics and some important ones. But this workshop, this webinar is the introduction for a workshop, which at the end I will talk a little bit about. Okay, what is non-invasive brain stimulation? So I think the technological advances in cognitive neuroscience introduced us with these techniques. And these are really important techniques because in theory and also in practice, they allow us, they give us a tool to study and modify brain function. Especially this modifying part is very important because then you can study the brain behavior relationship in a causal way. You can induce a change in the brain and then wait to see what happened in the behavior and cognition. There are basically two major techniques of non-invasive brain stimulation. The one that you see on the left side is TMS, transcranial magnetic stimulation, which is not our topic today, but briefly in TMS, there is a magnetic pulse through a coil, which at the end will change to electrical stimulation in the brain. And in the right side is TDCS, transcranial direct current stimulation, which is the most common tES technique at the moment. So tDcs sounds very easy. As you can see, there are two electrodes that you attach on the head or skull, and a very weak direct current is flowing between these two electrodes. And depending on where you put these electrodes on the head, you can modulate or modify brain function of different areas. Uh, this is one of the things that makes TDCS a little bit tricky, because everybody think it's very easy. It's also very cheap compared to TMS, so everyone can probably buy it and have it in the lab, but it's not that easy, you know, if you want to have good results in your research or good effects, therapeutic effects when you work with your patients. It's not that much easy. Uh, the important thing about these techniques that makes them very promising is that we can monitor cortical excitability and induce neuroplasticity. Cortical excitability and neuroplasticity, these are some basic physiological parameters of human brain and related to almost all of our cognitive functions and behavior. And importantly, especially the neuroplasticity is the part that is impaired in many neuropsychiatric disorders. That's why these techniques in theory and also in practice are very interesting to many people. There are also some people who don't like these techniques. I understand them, but in theory, I think these are very interesting techniques. And as I said, importantly, we can study brain behavior relationship in a causal manner. You know, fMRI and other neuroimaging techniques are very are older than transcranial electrical stimulation or TMS. But those techniques, which are very well documented, in best case scenario, they give us some correlational information about the brain, what happened in the brain when we are doing something. But with these techniques, we can induce a change in the brain and see the effects in the behavior. This is the big difference of these techniques. And the ideal thing is that if you could combine non-invasive brain stimulation and neuroimaging techniques, then you have the most cutting edge method that we have so far. Okay, our topic today is about transcranial electrical stimulation. We have three major TES techniques, and in this graph, I show you how they look. So in TDCS, as you can see, the top figure showing TDCS. Also a little bit talk about, you know, you see two colors, red and blue. The red means the target electrodes, or we could say also anodal, and the blue part on the right side is the reference electrode. We cannot say anodal or cathodal because in tACS and tRNS, in tACS especially, we cannot say we have anodal or cathodal. But anyway, in TDCS, we start the electrical stimulation. After a couple of seconds, normally 30 seconds, there is a ramping up in the stimulation, and you can see there is a direct current, electrical current, and then at the end, the stimulation is over. That's why it's called transcranial direct current stimulation, DCS. But we can change the way we deliver this stimulation. In tACS, instead of delivering the current directly, we can alternate. You can see this like a sinus, alternate the electrical stimulation and then deliver to the head. And this is called transcranial alternating current stimulation, ACS. And also we can randomly deliver these electrical currents to the head, which is called transcranial random noise stimulation, which we don't know still very well how it works, but there are some cognitive effects. This is just a brief overview, but today our topic is about TDCS mostly. Normally, this is how the devices look like. If you have seen these techniques, you know that mostly it comes two ways. The picture on the left is the classical TDCS, a classical TES, because with this device, mostly you are able to deliver both TDCS and TACS. But recently there has been some development, and which is called HD TDCS, high definition. So the idea behind this is to make a stimulation more focal by using smaller electrodes. This is just how the device looks like. I also got these graphs from PubMed 2 days ago, and just to show you uh, the interest in this technique. As you can see, from 2000, which was officially the year that TDCS was introduced, up to now, there has been more than 6,800 papers just in the PubMed. And you can see in the below graph that there is a yearly increase. Every year there is an increase in the number of published papers. And the same is true for tACS, although the number of published studies is quite low compared to tDCS, but I can say at the moment, tACS seems to be a bit hotter topic, I mean, compared to tDCS. And for sure in the future, we will see many more studies in tACS. So there is exponential growth in tDCS and tACS studies. And this is also a graph that shows interest in tES based on the Google trend. It shows how much these techniques were searched. So I don't want to talk about it too much because we don't have enough time. Okay, now we want to talk about tDCS, how it works, and the physiology of effects. This guy is Michael Nietzsche in 2000, when he introduced the current form of tDCS, because we know that tDCS has a long route, and also there are some other people who are working. So in the classical protocol, what Michael Nietzsche did was that these two figures on the right side are the early results of tDCS study. So he showed that with increasing stimulation, with anodal stimulation, we can increase cortical excitability, and with cathodal stimulation, we can decrease cortical excitability. So how we can say that, what does it mean, increase or decrease cortical excitability? Okay, the most common way to understand is to stimulate motor area, motor cortex, and then measuring muscle activity. So when muscle response are larger, we can say cortical excitability are enhanced. When they are decreased, we can say cortical excitability is inhibited. So these two graphs basically show, especially the top graph shows when the stimulation duration is longer, also the after effects is longer. But what I want to insist here is that this is the classic concept of TDC. It's not true that always cathodal TDCs decrease excitability, or always anodal stimulation increase it, especially when you deliver it for longer time. It's also important that you know a little bit about how we conclude about TDCS. About cortical excitability. As I said, just imagine you apply tDCS on your motor cortex, and then we measure motor evoked potentials. And the size of motor evoked potentials tells us about the effect of tDCS. We apply TMS over the motor cortex and then measure muscle activity from these muscles. In other labs, I think in most of the labs in the world, they use other muscles like the muscles here, but this muscle is called ADM. Anyway, from these muscles, we can record motor evoked potential. If you see, I have shown two MMP response here. The top one, which is marked by blue arrow, and the bottom one, which is marked by the red arrow. So when we say decreased cortical excitability, we means that this MMP response is smaller. As you can see, this is smaller compared to the below one. And when we say increased cortical excitability, this means that motor evoked potential size is larger. So far, most of the research, most of the common way for studying how TDCS works is based on the motor cortex. Why? Simply because we can see it's an objective way. With the size of these MMPs, we can conclude about the effects. But recently, there have been some other techniques that we can conclude about the other areas of the brain, like prefrontal cortex which is mostly studied with TMS EEG, which is not the topic here. But this is important to know because most of the studies that I want to talk about use these techniques for studying brain plasticity. Okay, and the physiology of TDCs is also something which is a bit complex. Also, we don't know exactly what happens. We have some assumptions, but basically what we know from animal studies and pharmacological studies, we know that glutamate, GABA, and NMDA receptors and calcium concentration, these are important physiological parameters behind TDCs. So in one of the early pharmacological studies, Michael Nietzsche in 2003, for example, they give participants in two studies, NMDA receptor antagonists, which blocks NMDA receptor, and also calcium channel blocker. So this means that when participants receive these medications, the NMDA receptors were blocked and also the calcium channel. And also there were two conditions, placebo, anodal TDCs and cathodal TDCs with placebo condition and with this medication. So they found that when these NMDA receptor antagonists and calcium channel blocker are taken, you can see that there is no MMP response. These white shapes in the middle of both graphs shows the medication condition. Instead, in the placebo condition, which participants take a placebo, you can see that anodal TDCs increased excitability and cathodal decreased. So this means that NMDA receptors and calcium channel, calcium concentration are important for TDCs effects. There's also cognitive effects of TDCs, which is personally more interesting to me because my field is cognitive neuroscience, but it is not the topic of today. But this is a recent review article that we published. I think if you go and see in this article, we discussed about how to study cognitive functions with both non-invasive brain stimulation and neuroimaging. But in the workshop that we will have the next 2 weeks, this will be discussed, especially for the clinical disorders. Okay, now I think from now on we want to talk about those factors that are important for us as a researcher to control or as a clinician to take care of. So let's see what are the factors that are important for the effects of TDCs, which also applies to other techniques like tACS. This paper was published in 2017, and in that paper, the authors shows some of the factors that affects our results. For example, history of muscle activity, time of day, lifestyle, age, gender, genetics, brain state. Some of these factors we have recently studied, the researchers have studied, but still some of them are not important. But the important point is that here is that these factors are important and affect our results. It's not easy just to apply TDCs or tACS on participants and expect some results without considering these factors. And today we want to talk about some of these factors. And also the below part of the graph shows those measurement factors or some stimulation parameter factors. And this is also another review article which I strongly recommend you to take a look. It's about the factors that determine variability of neurophysiological and behavioral effects in non-invasive brain stimulation, including TDCs. As you can see, there are some external or methodological factors. The methodological factors we can control as a researcher, and also some intrinsic factors which we might be able to control over, especially if we do some screening. Okay. And now the first group of factors that I want to talk, just some of them, is interindividual variability factor. So one of these interindividual difference is anatomical difference. You know, all of us have different brain. My brain is different with your brain. We have different skull. In my experience with a lot of participants during my PhD, I noticed that, for example, with some of them, you can never find a good motor evoked potential. It doesn't mean that the brain doesn't work or is not good enough. It's just anatomical difference. As you can see, there is scalp, there is uh, cerebral fluid, there is gray matter, white matter, and there is anatomical difference. This paper in 2015 investigated intersubject variability in electrical fields of motor cortical tDcs. And I think this part is very important. Here it says, cerebrospinal fluid thickness explain 1/2 of the intersubject variability in the electrical field. So this is something that we cannot control because it's for everyone is different, but it's important to know that these anatomical differences are important in our results. This is also a graph that we, from our paper in 2020. So here we modeled one stimulation protocol over the head, in the head of an adult on the left side and a children, a 10-year-old children on the right side. So as you can see on the left side, this protocol was 2 milliampere with the same electrode size, and the induced electrical field was 0.8 with 2 milliampere. So when a similar stimulation protocol was applied, except that here the stimulation intensity was 1 milliampere, which means half of the protocol used in the adults. So here the maximum electrical field was 0.6. So if this means that you cannot transfer the results from an adult brain to the children brain, because if this was the case, here the maximum field should have been around 0.4, half of 2 milliampere. And what is the take-home message here is that if you work with participants of different age, you should consider the age. Otherwise, this can affect your results. Another inter-individual factors that we can control and should is aging. This study was done in 2012 by one of our lab members, and in this study, the experimenter recruited 3 groups of participants: young participants, pre-elderly participant, which is marked by circle, and elderly participant, which is marked by triangle. So, and she applied the tDcs for 1 milliampere and 15 minutes, and then examined the motor evoked potential, the motor cortical activity. As you can see, the elderly group, which is marked by triangle, they showed no motor evoked potential after anodal tDcs. Although for cathodal tDcs they showed, but still the level of plasticity induced in the young and pre-elderly group was significantly larger. So here the take-home message is that with aging, the brain plasticity will reduce. So if you want to have an experiment, you should consider age. You cannot have 10 young participants, 10 mid-age participants, and 10 old participants, and then expect a homogeneous results. This is another graph from another lab members that again investigated the effect of aging. Here again, in this experiment, different protocols of tDcs, 1 milliampere, 2 milliampere, 3 milliampere, with different durations, 15, 20, and 30 minutes were examined. And I think the take-home message here is that only 3 milliampere could induce long-term excitability in all groups, especially in the old groups. But again, the point of this graph, I know there is a lot of information here. The point of this graph is that there is reduced plasticity in the brain with aging. So aging is another factor that we should control, at least in the experimental design. Another inter-individual factors is chronotype. So this was part of my PhD thesis, as I explained in the beginning. In my PhD thesis, I studied the impact of chronotype and sleep pressure. So the article out of this thesis, after a very long peer review and painful peer review, came out a few weeks ago. Chronotype, what is chronotype? Okay, chronotype is an individual difference in circadian preference. Most of us have a trend to be a morning type or evening type. I'm personally evening type, and majority of people are neutral, but still have some trends toward morning or evening time. And you can easily determine you are morning or evening type. There is a questionnaire. It has 12. Sorry, it has 20 questionnaire. If you fill, you can see if you are morning or evening type. This red circle shows the sleep onset and wake up time for those who are evening type, and the blue circle shows roughly the sleep onset and wake up for morning type. So if your sleep timing falls within these circles, then you can see if you're morning or evening time. Okay, what we did in this experiment, we recruited two groups of people, one group of early chronotypes and one group of late chronotypes. And we applied anodal TDCS, cathodal and sham TDCS in the morning and also in the evening. Again, with anodal TDCS, we expect to see increased excitability or increased MMP size. With cathodal, we expect to see decreased MMP size. And then we measured after effects, these motor evoked potentials with TMS and EMG electrode. So this is the results. These results clearly show this is for early chronotypes who are morning types. These people showed significantly larger plasticity in the morning after anodal and cathodal TDCS. But the important thing is that they still showed plasticity in the evening, but the size, as you can see, the size was significantly lower. And the same was true for late chronotypes. They showed significantly larger plasticity in the evening after anodal and cathodal TDCS, although they also showed significantly showed some plasticity induction in the morning. But the important thing is that the size was significantly larger. The effect size was significantly larger in the evening. So just imagine that you have an experiment, or you have some group of patients, and these groups, some of them are morning type and some of them are evening type, and then you apply your intervention or your protocol at the wrong time. So then if you don't have good enough results, this simply could be due to bad timing of the day. Okay, so the interindividual factors, I just talked about anatomical, age, and chronotype. There are different factors which we cannot cover at the moment. The other important group of factors that I want to talk is the stimulation parameters, because I think this is where we as a researcher and clinician can have most influence. We can decide which factor to modify which factor or to change the stimulation parameters in the serve of our expected effects. Okay, the first thing that I want to talk about the stimulation parameter is electrode placement. First of all, there are different electrode size. As you can see here, normally the most common used size is 35 centimeter square centimeter. But and these rubber electrodes are usually covered with a sponge. So this is also important, how wet and how dry should be a sponge. We can use saline solution or 1020 paste for some other gel for doing conductance. But in recent years, for the HDTDCS, smaller electrodes have been introduced. And the idea behind this electrode is to make the stimulation focal. But the important thing is that here for this slide is that we have different electrodes and we should choose the best size for our experiment. You cannot use large electrodes for your participants if they are children, or vice versa, or you cannot have electrodes too close together, which we talk again soon. One important thing that you should control is how you can find your target region. Okay, maybe you want to stimulate left dorsolateral prefrontal cortex, which is F3 according to 1020 AG system. How do you determine this region? I know most people use some meter and some measurement, but you have to make sure that these are accurate. You cannot, even couple of not centimeter. I mean, the millimeter is fine, but sometimes if you don't put the electrode on the right target, you cannot see clear effects. It's very important. These are some of the basic things that I know in most lab it's not very done accurately. So the most common way of targeting is using 1020 EEG system, which you should practice enough in order to be expert in finding target region. But methodologically, it is suggested to use neuronavigation, which means you ask participant, you have the MRI data of participant, and based on the MRI data or the imaging brain of the participant, you will determine your target position. But we know that this is not doable in every lab because it's very expensive setup. You should have MRI, you should have navigation. Also, methodologically, it's a bit complicated. But you have to try to, at least when you use a standard EEG system, to do it correctly. Another important thing that probably is not taken into account is electrode placement, the angle. This study was published in 2019 by one of our lab members. In this study, they compared, okay, what happened if we put target electrode in 45-degree angle or without any angle. So this the middle head, which is marked by this red square, is where the target electrode was with 45-degree. So as you can see in the figure below, when there was 45-degree angle, only in these cases there was induced plasticity after anodal Tdcs and cathodal Tdcs. And I know in most labs, this is not in the motor cortex. It's important. This is for the motor cortex. I know in most lab the target electrode is not without any angle. So this is very basic things and very easy thing that you can control. But if you don't know the effect, probably this will affect your results. In addition to angle, the size and distance of electrode is also important. Again, this graph is the modeling of the electrical field in a head of a children, 10-year-old children, but in the left side, the electrode size was 5 × 5, and in the right side was 5 × 7. So which means with larger electrode, the electrodes are very close together. There is no distance enough. So and with the first protocol, the stimulation intensity was 1 milliampere. With the second protocol, with larger electrodes, the stimulation intensity was 2 milliampere. So if you now look at the electrical field, you can see that the induced electrical field from 1 milliampere was almost like 2 milliampere. So what happened here? The reason is that because the electrodes were too close together, a lot of direct current were shunting down, shunting down. But if there was enough space or distance between these two electrodes, this electrical field should have been larger for the 2 milliampere, almost double of this, but this didn't happen. So it's also important to consider the distance between electrodes, especially if your target regions are close together. So this is some basic take-home message. The electrodes should not be too tight, not too loose, not too wet, not too dry, always in a constant position, and not too also close together. At least from experimental point of view, these are something that we can control. Okay, another stimulation parameter. I don't know why this red line appear in my screen. If you can see it also, I didn't do anything anyway. So another stimulation parameter, two important ones that we can also control, is the stimulation intensity and duration. There is a misconception. I cannot say misconception. There is something that a claim that is not always true. We believe that larger or stronger stimulation resulted in better effects or larger effects or long-term effects. And also the longer stimulation, for example, 20 minutes of stimulation compared to 5 minutes, resulted in longer after effects. This can be true in some cases, but cannot be true in all cases. So in this study, which is done by another lab members in our lab in 2020, he investigated the impact of 1 milliampere, 2 milliampere, and 3 milliampere stimulation with different duration, 15, 20, and 30 minutes. And the major result was that uh, only the 3 milliampere stimulation, um, not only the 3 milliampere stimulation, uh, all of the stimulation could induce excitability up to the next evening. You know, he measured excitability up to 30 minutes, then up to 2 hours, then up to the same evening and next evening. This SE means same evening and NE means next evening. So in all of the intensities, you can see that especially the 20-minute duration could induce excitability up to the day after. Sorry, only 2 milliampere and 3 milliampere, because this triangle is colored only 2 milliampere and 3 milliampere. Only with the higher intensity, he could induce excitability up to the tomorrow, which is important for clinical application. But again, the difference induced by 20 minutes with 3 milliampere was larger. Okay, this is for anodal case, for anodal stimulation. With anodal stimulation, we can say yes, with larger stimulation, you can induce larger effect, but not necessarily with longer stimulation, because here 20-minute stimulation was more effective than 30-minute. This is for anodal. But another lab members did the same thing for cathodal stimulation. So basically, he investigated 1 milliampere, 2 milliampere, and 3 milliampere. This study was done by Mosaibi, Salmani et al. And what he found, actually, he found that when he applied 1 milliampere and 3 milliampere after cathodal stimulation, the expected effect appears. What is the expected effect after cathodal stimulation? It is to reduce the size of MMP or induce long-term depression-like plasticity. As you can see, the motor evoked potential in the first graph and in the below graph are reduced. But after 2 milliampere, there is an increase. There is an increase in the motor evoked potential. So this was first confirmed in a study in 2013 by Batsit Katse. He was also another PhD student of Michael Nietzsche, but now you can see the results is confirmed in this study. So the take-home message here is that if you want to apply cathodal tDcs to reduce something, to reduce excitability or to reduce one function, but you cannot achieve it with 2 milliampere, at least in the motor cortex. So cathodal stimulation behavior is different. In the anodal stimulation, yes, with increasing intensity, you can expect larger effects, but for cathodal, this is not true. Okay, the summary for these two parts is that there is dosage-dependent TDCS effects, and there is not a linear relationship, especially for cathodal TDCS. And TDCS beyond 1 milliampere and also beyond 15 minutes can gradually improve efficacy. So this one probably is confirms our conception that, okay, TDCS longer and larger TDCS with stronger intensity and longer duration is more effective. But this is again mostly true for anodal TDCS. Also consider that these factors and findings are studied for motor cortex. Maybe this is not true for the prefrontal cortex. I think there will be 2 papers published soon from our lab that in those papers the same investigators did the same thing for prefrontal cortex with TMS EEG, and as far as I know, the results were somehow the same, especially for anodal stimulation. Okay, there are other stimulation parameters which I cannot cover now. What is the role of stimulation repetition? How many times we should repeat, especially for clinical purpose? What should be the time, the interval between stimulation sessions, 24 hours, 72 hours, or 1 week, especially for clinical? What is the difference between online and offline stimulation? Just briefly explain that online stimulation means that when we stimulate and the participant is doing something, for example, doing a task concurrently with the stimulation, and offline stimulation is that we stimulation first and then do the task or the test. What target region should be used if we want to, for example, target F3, left dorsolateral prefrontal cortex. Should we put the electrode exactly on F3? This might not be true because the modeling results sometimes suggest this is not true. What is the role of reference electrode position? Because we know that reference electrode position is mostly cathodal, but many researchers don't care about the role of cathodal stimulation. They just think that it is a reference electrode. As I showed you, 2 milliampere cathodal can change the stimulation polarity. What is the stimulation polarity? It should be anodal or cathodal. How we can make stimulation focal? What is about the multi-channel stimulation protocol, which is the new suggested protocol? And also there have been recent development about concurrent tES fMRI. Unfortunately, we cannot cover this, but these covers will be discussed in the workshops, hopefully. Okay, now we talk about some factors that we can control and screen in the experiment. There are some factors that previous studies have shown that has effects on excitability. I will show the details of these factors and the studies later in the workshop, but we know that, for example, smoking is important. If you want to have an experiment, you should exclude smokers, especially if your target region is motor cortex. Or caffeine consumption. We cannot exclude this because almost everyone uses caffeine, but you should, as participants, not to drink coffee or whatever has caffeine, like energy drinks, at least 1 hour or 2 hours before the experiment. Or physical exercise. Here in our own lab, when participants come to our lab with bicycle or running, because many young people do this, we cannot start experiment right after. We ask them to sit relaxed at least for 30 minutes because this affects excitability. Whatever makes your brain state altered, like a stressful event. I had a participant in one of my experiments, and her results was very, very good. I mean, very easy. You could find the hotspot, you could see the motor evoked potential. And in one day, it was completely, there was no effect. And then, for example, I found that tomorrow she had an important exam. So you should consider this when you have your experiment or you have your clinical intervention. Sleep pressure, which I talk soon, because this is part of my PhD thesis, and chronotype. As I said, chronotype, you can easily screen persons with chronotype. It means that just give the participants or your patient that questionnaire and apply your intervention at the right time. Okay, about sleep pressure, this is a second part of my thesis. This is one of the factors that we can also screen and control. In the second part of my thesis, I did the same experiment, but not in the chronotype, in two different conditions. After having a normal sleep, which is marked by NS in the figure, or after having a sleep deprivation. So we asked, I had 30 participants and asked them to have normal sleep overnight at their home and then come to the lab. And in the next session, they come to our lab and did not sleep the whole night, which was very difficult. And then we applied TDCS, anodal and cathodal TDCS, and then measured the after effects. This paper is currently under revision, but you can see the results for more details as a preprint. So this is the results of anodal TDCS. So when they had normal and sufficient sleep, you can see that anodal TDCS could induce excitability, like MMP is increased. But interestingly, after sleep deprivation, there is no effect at all. It's like sham stimulation for the same participant. So this is very important. Sometimes we don't see any effects at the end because we don't simply control sleep pressure. But the results of cathodal is much more interesting to me. After cathodal TDCS, when the participant have sufficient sleep, you can see that cathodal TDCS could induce MMP, LTP like MMP. But when there was sleep deprivation, there was a sleep deprivation, you can see the cathodal effects are turned into anodal-like. This is also very surprising to us. We didn't expect such thing, but I think these are important to know that all of these factors can affect your results, and simply you can control some of these factors. Okay, and in the last part of the experiment, in the last part of the talk, I want to talk about some experimental aspects that we can control, like experimental design, what is the best control condition, sample size, and outcome measure. We have different design like crossover and parallel group. In the crossover design, we recruit one group of participants and assign them to different intervention like treatment A, treatment B, and also a control condition. And also we have parallel group trial, which means, for example, we have 3 separate groups and each group receive 1 intervention. So for clinical purposes, especially if you want to measure clinical efficacy, using parallel group is much more beneficial. Of course, it needs more time, and also you have to spend more resources because you need 3 groups of people. But again, about the benefits and of each of these design, we will talk later. But you need to know which design is good for you. I know some people only use pre-post design. This is not really good design, as not acceptable in the current in nowadays. If you want to publish a result, definitely with pre and post result, you cannot publish the results in a good journal. But you need to know what is the benefits of one over and choose the best design. For example, for crossover, if you have 3 sessions and in each session you apply the same task, you have to make sure that the task is not the same. You use different script for each task, because then there may be practice effect. For example, last session, the effect could be larger, or you should make your stimulation session counterbalance or randomized. So these are the things that you should consider. As I said, another thing about the design is to have especially follow-up design, pre, post, and follow-up. This is especially important for clinical, for those who have clinical interest and want to examine clinical efficacy. And also, I recommend that always use trial registration. Try to register your work as a clinical trial, because this will make your work much more stronger. You have to do some evaluation. Some people review your work, and then before you start your work, you know some of the weakness and advantages of your works. And another aspect that we should control is control condition. But the question is, what is the best control condition? In tDCs and tES research, we usually have sham condition as a control condition. But again, it's important to consider how to apply sham condition. Do it as in a crossover design. One of the stimulation condition is sham condition. In a parallel group, one group only receives sham stimulation. But some people like to use baseline control. Just do a baseline measurement and then intervention, and then again do the same measurement in one group. This is not really good, and it's very weak control condition. The best thing is to have a sham condition, and especially if you have a control group, one of your control group should receive sham stimulation. Because some of us don't like to have a group with sham stimulation. We think it's waste of time. It's not waste of time. It's actually, it is important to make your work reliable. About sample size, this is another factor which has been criticized recently, because a lot of studies in the last decade were published with low sample size. I understand in some cases you cannot have a lot of samples, like for example, autism is one of the areas that they work. You can hardly find a lot of autistic children with the criteria that you need, but always use power analysis for your research. For example, in this slide, it shows that if you have two groups and you have 4 measurements, for example, pre, post, and 2 follow-ups, you need 36 participants, which means 18 participants in each group. And you have 3 measurements, then you need more than 40, you need around 50 participants. For at least have 15 participants per group if you have more groups. Otherwise, it's very difficult then to make you cannot expect people trust your results. And this paper I also strongly suggest you to take a look, because it specifically talks about the importance of sample size for reproducibility of tDCs effects. And the last experimental aspects that I want to talk is about the outcome measure. Okay, why we use tDCs or TACs or TMS? because we want to measure the effect on something. But what measure we should use? If you have only one measure and that measure is only behavioral, then this is not, then you cannot understand what is the effect. You should have different measures, preferably from behavioral performance, from physiological measures, and some cognitive measures. For physiological measures, we can use EEG and some imaging methods like fMRI, but I know that these are very costly methods. But the whole point here is that you should have more than one measure in order to, if you want to trust your results. And sometimes it also helps you because you don't see any effects on the behavioral performance. For example, you apply tDcs for improving working memory, you don't see any effects at the behavior, but you see significant changes in the EEG parameters like brain oscillations. Then you can have some explanations. So we should try to have outcome measure from basic physiology, hopefully to behavioral cognition. And I know some people only use some questionnaires, and which is not really acceptable at this time anymore. Of course, for some clinical patients, you should measure the symptoms with questionnaire, but you should also have some objective way to measure your effects. And the last, this is something that I don't want to talk because time is over, but we'll discuss this for sure in the workshop. How we can maximize the effect, especially for clinical application. How strong should the stimulation be? How long should the stimulation be? How many sessions? Can we also combine with other intervention, like for example psychotherapy, some kind of training, or if we can, how it should be? Which one should come first? What are the suggested promising protocols? There has been recently a paper that tried to introduce the promising and suggested protocols based on the group of experts. And in the workshop, I will discuss that. And also application of TDCs in pediatric publication, which is also among my interest. So these topics, so hopefully we can, for sure, we will discuss this in the workshop. And there is also some open questions and topics that unfortunately we could not cover today, maybe in the future webinars, about optimizing the protocol, multi-channel stimulation protocol, which is the current trend, how to focalize stimulation, how to combine protocols, what are the best blending strategies, why they are important. Can we transfer our results from motor to non-motor areas? Because I know many of us, including you, are interested in cognitive areas. Like prefrontal cortex is the most common target region for in clinical applications, and also about tES protocols. And again, these topics will be discussed in the workshop. And the last slide is slide of today's talk is about the current trends. What are the current trends? If you want to stay in this field, what are the current trends that you should focus? One trend is optimization of a stimulation protocol. How we can optimize your stimulation protocol to have larger and more effective effects. And the other thing is individualizing your protocol. If you remember in the first slide, I showed you that there are different anatomical difference between me and you and everyone. So now the idea is that, okay, use the individual MRI and induce your stimulation and arrange your stimulation parameter based on each individual anatomy. For example, maybe for my head, 2 milliampere would be enough. Maybe, for example, for someone, 1.5 milliampere leads you to the same results. And another very interesting approach is multimodal approach, and to combine tES with imaging techniques like fMRI and EEG. If you want to stay in this field, like these are the trends that you can go on with. And this is about the workshop, which will be in 12th of September. In this workshop, we will talk about basic of tDcs. These parts highlighted in the yellow is the topics that we did not talk today, but we will talk later. About methodological consideration, we talk about what we talked today in more details, also with some other parameters, and also some majority part of the workshop will be about clinical application, because this is my interesting, my area which I work a lot. And also we show you some hands-on TDCs, how to work with TDCs. Thank you very much for your time, and sorry if some of you could not join. but I recorded this webinar and we can send later to everyone. Now I'm open to your questions. Yes, they ask, can you briefly define how to use power analysis? I think if I can share my desktop, then I can show you. Let me open my G power. Okay, can you see my G power on the left side? Okay, just imagine that we want to use, we have, normally most of our designs go with F tests. We usually have between subject and within subject factor. Let's imagine that we want to have what I did in my own experiment. I had two groups of patients, two groups of participants, late chronotypes and early chronotypes. So this is a between subject factor, different group. And they did the same measurement in different thing, in different days. This is within subject factor. Okay. How many groups I have? I have two groups. How many measurements I have? So TDCS, it was 6 sessions. Actually 3 sessions because each group attended 3 sessions. But I had also 2 sessions for cognitive tasks and cortical excitability, which make it 5. This effect size is different. Normally the default is 0.25, but you can change it, but I will keep it the same. So with this effect size, it tells me, okay, if you want to have a power of with a power of 95 and alpha of 0.5 and with the effect size of around 25, which is the like medium, you need to have 32 patients in total, 32 participants in total, which was the case for me also, which means 16 16 group in each group. You can change these parameters, but I think in that paper that I introduced you, it told you you should do it with the medium effect size. Of course, this effect size, you should also use the right effect size, because in the mixed model ANOVA, your effect size is partial eta square, not like F. But there are a lot of great videos in the YouTube. If you search about G power, you can determine your sample. But it's important to know that for correlational studies, you need much more larger sample size. This is experimental design. For correlational, you really need more sample size. Yeah, software G power. This is, I will write it in the chat. It's G power software. It's a free software you can easily download. I chat to the host, and now I chat to everyone. G power. You're welcome. Yes, it's recorded, and I asked the host to record it, and I think hopefully they can send you the file and give you the link to access the file. All right. I know that many of you probably didn't have enough, didn't have prior experience maybe with these techniques. I tried to be a little bit basic in the way I explain, but in the workshop that we will have, it will be more detailed, and there will be also some practical aspects of TDCS and tES. Oh, there's another question. If we use tDCs with EEG, how we will differentiate brain potentials from electrical stimulation. How can we prevent ectopic electrical signal? Okay, this is one of the things that we did not discuss today about tES and EEG, which is one of the current trends. So, this is actually one of the challenging part, because we in our lab we are doing some tES, EEG stimulation. So you can design a script or for EEG analysis to get some data, but in our experience we could not use the data from the concurrent stimulation and the EEG. But after the stimulation period, definitely you can see the change in the brain oscillation. So basically you should have a resting state measurement before the stimulation and a resting state measurement after the stimulation. So when comparing these two resting stimulation, then you can differentiate between the brain potentials, I think brain oscillations after the intervention. But during the stimulation with tDCS, it's a little bit noisy because it's a direct current, but with tACS, because there is alternating stimulation, there may be some way, which at the moment is not our topic, but the question was how to differentiate. In order to differentiate, you should have a reliable baseline before your intervention, and then apply tDCS with the EEG, and after the intervention, again, measure the resting state. I think, yes, the science team says that they will share a video with you, and they ask you about the workshop. Again, I think they will be touching with you. All right, if there is no question, I just say goodbye and thank you for participating. I'm sorry if some of you had problems in joining, and hopefully we can see some of you in the workshops if there is no more questions. Thank you. Have a good day.